Hi, welcome to our channel of IGNOU audiobooks, Indira Gandhi National Open University, School of Agriculture, SOA, Master's Degree Programs, Master of Science in Food Safety and Quality Management, MSCFSQM, First Year, MVPI 001 Food Microbiology, Block-1 Fundamentals of Food Microbiology, Unit-4 Beneficial Role of Microorganisms 4.0 Objectives. After reading this unit, we shall be able to describe beneficial role of microorganisms, explain fermentation, its science and technology, comprehend the needs and types of food fermentations, state common examples of different types of food fermentations, and attribute fermented foods as functional foods. 4.1 Introduction. Food microbiology can be divided into three focus areas: beneficial microorganisms, spoilage microorganisms, and disease-causing microorganisms. Beneficial microorganisms are those used in food fermentation to produce products such as cheese, fermented meat, pepperoni, fermented vegetables, pickles, fermented dairy products, yogurt, and ethnic fermented products such as sauerkraut, idli, and kimchi. In fermented products, produced by natural or controlled fermentation, microorganisms metabolize complex substrates to produce enzymes, flavor, compounds, acids, and antimicrobial agents to improve product shelf life and to prevent growth of pathogens and to provide product attributes. Microorganisms, with their enzymes, also break down indigestible compounds to make the product more palatable and easy to digest. In addition, the beneficial microorganisms also serve as probiotics to impart direct health benefit by modulating the immune system to provide protection against chronic metabolic diseases, bacterial infection, atherosclerosis, and allergic responses. Examples of beneficial microorganisms are Lactobacillus acidophilus, Lactobacillus arabinosus, Lactobacillus lactis, and Pediococcus cerevisiae. Food spoilage microorganisms are those which, upon growth in a food, produce undesirable flavor, odor, texture, and appearance, and make food unsuitable for human consumption. Sometimes, uncontrolled growth of many of the beneficial microorganisms can also cause spoilage. Food spoilage is a serious issue in developing countries because of inadequate processing and refrigeration facilities. Examples of food spoilage microorganisms are Brocotrix, Lactobacillus, Bacillus, Pseudomonas spp, and some molds. The microenvironment created in a spoiled food generally discourages the growth of the pathogenic microorganisms, which are considered poor competitors. 4.2 Fermentation. The uniqueness of several microorganisms and their often unpredictable nature and biosynthetic capabilities in a specific set of environmental conditions have made them ideal candidates. In attempts to solve difficult problems in life sciences and other fields, microorganisms have been used in various ways over the past many decades to advance medical technology, human and animal health, food processing, food safety and quality, genetic engineering, environmental protection, and agricultural biotechnology. The use of beneficial microorganisms in the food sector has been a long tradition, namely lactic acid bacteria and yeasts in fermentation processes. The former are widely used in the manufacture of fermented food and are among the best studied microorganisms. The fermentations may be by yeasts, bacteria, molds, or combination of these organisms. Detailed knowledge of a number of physiological traits has opened novel potential applications for these organisms in the food industry, while other traits might be specifically beneficial for human health. The food and beverage industry exploits non-pathogenic microorganisms for the production of fermented foods. These foods are prepared from raw materials and acquire their characteristic properties by a process that involves microorganisms, in certain cases the endogenous enzymes of the wild microflora nature to the raw material may play a decisive role. It is believed that fermented foods originated from the Orient and date back to the prehistoric times. Originally, these were fermented spontaneously by autochthonous strains found in the raw materials or the environment. This was the start of traditional biotechnology. The most important were cheese, yogurt, wine, vinegar, beer, bread, and the traditional fungal fermentations used in Asia and Africa for the production of food. In the biochemical sense, the term fermentation refers to a metabolic process in which organic compounds, particularly carbohydrates, are broken down to release energy without the involvement of a terminal electron acceptor such as oxygen. Partial oxidation of the substrate occurs so that only a relatively small amount of ATP energy is released compared to the energy generated if a terminal electron acceptor is involved. Partial oxidation of a carbohydrate can give rise to a variety of organic compounds such as alcohols, organic acids, and acetone. The compounds produced by microorganisms vary from organism to organism and are produced through different metabolic pathways. The term fermentation can also be applied to any industrial process that produces a material that is useful to humans, and if the process depends on the activity of one or more microorganisms, consortia. These processes, known as industrial fermentations, are usually carried out on a large scale and in vessels in which the organisms are normally grown in liquid media. Some industrial fermentations are fermentation in the biochemical sense, but the majority of microorganisms involved are aerobic and use oxygen as terminal electron acceptor and thereby metabolize carbohydrates completely. A vast range of materials are produced by industrial fermentations. These include 1. Organic chemicals used as fuels, food additives, antibiotics and enzymes for use in the food and other industries. Vinegar is an example of a food additive produced by an industrial fermentation. 2. Organisms are produced on a large scale for the extraction of protein, single-cell protein, that can be used as a part of the human diet. Corn is an example of a single-cell protein. It is produced from the fungus Fusarium graminearum. This mycoprotein, purified from the fungus, is currently available for use as a food and is incorporated into a range of dishes that appear on supermarket shelves. Meatless dishes with a high protein content made from corn are particularly appealing to vegetarians. 3. Yeast cells produced for use in industries such as the baking industry, which relies on the mass production of large amounts of baker's yeast. 4. Foods produced on a large scale as a result of the activities of microorganisms, e.g. cheese, yogurt and bread. 5. Production of alcoholic beverages, e.g. beer and wines. 6. Cellular extracts used as food additives, example, yeast extracts from yeast cells, produced as a byproduct of the brewing industry. 7. Industrial fermentations are now often considered under the heading of biotechnology, i.e. technology that uses living organisms and their products in the manufacturing and service industries. 4.3 Fermented foods and their importance. Fermented foods are those foods produced by the modification of a raw material of either animal or vegetable origin by the activities of microorganisms. Bacteria, yeast and molds can be used to produce a diverse range of products that differ in flavor, texture and stability from the original raw material. The production of many fermented foods involves organisms that are biochemically fermentative. Lactic acid bacteria that ferment carbohydrates to produce lactic acid are particularly important, but yeasts also play a major role in some food fermentations, fermenting carbohydrates to produce ethanol and other organic chemicals. Molds that do not ferment carbohydrates also play an essential role in some food fermentations, for example, the production of cheeses, blue cheeses, and oriental foods, soy sauce. Fermented foods are an extremely valuable addition to the human diet for a whole variety of reasons. 1. Increase in variety. Fermented foods increase the variety of foods that are available, adding to our diet a group of highly nutritious products with unique characteristics. There are, for example, about 1,000 different types of cheeses. 2. Use of ingredients. Fermented foods form an important ingredient for a wide variety of dishes and are often used to impart special flavors. Example, pepperoni in pizzas, yogurt in curries, cheeses in a whole range of dishes, including soups, and soy sauce in stir-fry dishes. 3. Improvement in nutritional quality. The fermentation process may improve the nutritional quality of a raw material. Here are some examples. Times tempeh fermentation raises the vitamin B12 content of the original soybean. Times tapioca fermentation doubles the protein content of cassava and increases the level of essential amino acids. Times the presence of yeasts in a fermented food will increase the vitamin B content. Times antinutritional factors such as phytates, glucosinolates, and lectins may be removed by the fermentation process. Times fermentation may lead to an increase in the bioavailability of minerals. These improvements in the nutritional value of raw material will have little effect in the balanced diets of Western populations. However, for populations that subsist on diets consisting largely of polished rice, maize, or other starches, such as in Africa and Asia, the contribution that fermented foods make to the intake of B group vitamins and proteins is highly significant. 4. Preservation. Fermentation often preserves a raw material, improving safety with regard to foodborne pathogens and increasing shelf life. Compare the shelf life of raw milk, only a few days, with the shelf life of yogurt, several weeks. 5. Health benefits. Some fermented foods are said to have definite health benefits, although the scientific evidence for this is limited. Reports suggest that fermented milk products such as yogurt can reduce serum cholesterol levels and help avoid cancers, particularly those associated with the colon. Bio yogurts, AB and ABT yogurts, are said to have a restorative effect on a normal microflora, assisting recovery of a normal balanced flora after oral antibiotic therapy. 6. Improved digestibility. Some fermented foods are more easily digested than the original raw material. People who cannot digest lactose properly, show lactose intolerance, can often consume some types of fermented dairy products, particularly yogurts, without harmful effects. Lactose intolerance is due to the absence of the enzyme galactosidase in digestive juices, which converts lactose to glucose and galactose. Ingestion of dairy products leaves unabsorbed lactose in the gut, which is fermented by the normal gut flora, giving flatulence, abdominal pain, and diarrhea. The fermentation of milk converts the difficult-to-digest lactose to the more easily digested lactate, and the galactosidase in live starter culture organisms appears to assist in the digestion of any residual lactose. Legumes, e.g. soybean, contain oligosaccharides such as stachyose, which are fermented in the gut to yield gas and the associated socially embarrassing flatus. The oligosaccharides are broken down to readily digestible monosaccharides and disaccharides during fermentation of legumes by molds, thus removing the problem. Fig. 4.1 Functional properties of starter cultures for food fermentation. 7. Detoxification of raw materials. the fermentation process may remove toxic chemicals present in the raw material. Cassava fermentation, for example, removes a cyanogenic, cyanide-producing glycoside. Cassava is toxic if eaten raw. 4.4 Food fermentation, science and technology. The science of fermentation is known as zymology. Food fermentation involves all those fermentation processes where either the ultimate product is used directly as a food, as an additive to food, or is a basic ingredient to the food, or the byproduct formed during fermentation, food waste utilization, their disposal or proper management. As a science, the food fermentation has an element of biological sciences, especially the microbiology, genetics and biochemistry. As a technology, the food technology, chemical engineering, along with integral component of sciences involved in food toxicity, acceptability and food nutrition. For any process to make fermented food, essential components are raw materials, microorganism, starter cultures, fermentation vessel and associated controls, processing, recovery and packing systems. But from fermentation technology point of view, the fermenter and microorganism involved assumes prime importance. Since the end product would serve as a food, it is essential to evaluate it from nutritional, toxicology and sensory quality point of view, and thus, the knowledge of these aspects would form an integral part of food fermentation technology. The technological dimensions of food fermentation towards the application of engineering sciences in designing the fermenters and associated controls for optimum fermentation and product recovery are immense. As the product on the commercial scale would be marketed, which can occur only if it is economical, and therefore the economics of such products and the associated marketing aspects cannot be ignored. Similarly, transfer of the knowledge to the scale of technology operation required is of paramount importance. The scale-up operations, harvesting, biocatalysis of microorganisms, product recovery, effluent treatment, etc. are of concern to fermentation technologists. Food fermentation technology could be organized based either on the product or the system used to make the fermented foods or products derived from it, similar to food biotechnology. Refer Fig. 4.2, Fig. 4.2, a generalized scheme of various operations employed in food fermentation technology. Look at the screen. 4.5 Types of food fermentations. A number of different types of food fermentation can be recognized. 4.5.1 Acid food fermentation. These include acid fermented dairy products, e.g. cheese, butter, yogurt and kefir, acid fermented vegetable products, e.g. sauerkraut, olives and various pickles, acid fermented meat products, e.g. the semi-dry fermented meats such as cervelat, and the dry fermented meats such as salami and pepperoni, sourdough breads. The common feature of all these products is the use of lactic acid bacteria to carry out the basic fermentation process. Modern production systems usually involves the use of starter cultures. An exception is the fermentation of sauerkraut, for which the process depends on lactic acid bacteria that are natural inhabitants of the surface of cabbage leaves. Sometimes sugar is added to raw material to allow the lactic acid bacteria to produce sufficient acid for a successful fermentation. This is the case with fermented meats, in which the sugar content of the raw material is very low. Salt may be added to suppress the growth of the normal spoilage microflora and allow the lactic acid bacteria to dominate, e.g. sauerkraut, pickles and fermented meats. The raw materials may be pasteurized to eliminate pathogens and suppress natural contaminants that compete with the lactic acid bacteria used in the starter culture. 4.5.2 Yeast fermentation. Yeasts are important in food fermentation because of their ability to produce carbon dioxide and ethanol. Carbon dioxide is the important metabolic product in the manufacture of leavened bread, whereas ethanol is metabolized in the production of beer, wine and spirits. 4.5.3 Solid-state fermentation. Solid-state fermentation involves the use of a solid substrate into which the fermenting organism is inoculated. The organisms used are often molds. Examples are the koji process and the second stage of tempeh fermentation. 4.6 Common examples of food fermentations. A very wide range of innumerable products of the food industry, such as sour cream, yogurt, cheese, fermented meat, bread and other bakery products, alcoholic beverages, vinegar, fermented vegetables and pickles, etc., are produced through microbial fermentation processes. The efficiency of the strains of the organisms used and the processes are being continuously improved to market quality products at more reasonable costs. 4.6.1 Oriental and indigenous fermented foods. A large number of fermented foods can be grouped under the heading of Oriental and indigenous fermented foods. Fermented foods of this type are produced in Asia and Africa and are often associated with specific countries or areas. Most of the products are unknown in the West but frequently have major nutritional role in the diets of the local population. Lactic acid bacteria are involved in some of the fermentation, but yeast and molds are often the main organisms responsible. Many are solid-state fermentations or involve fermentations of more than one type. Some of the products are manufactured on large scale, but many are carried out on a cottage industry or household basis. The major groups of these products, with example as are given below. Table 4.1 Oriental and indigenous fermented foods. Look at the screen. 4.6.2 Fermented vegetable foods. A. Sauerkraut. Sauerkraut is fermented fresh cabbage product. It is popular in USSR and Europe. The main organism involved in the fermentation of this pickle is lactic acid bacteria, Leuconostoc mesenteroids, followed by Lactobacillus plantarum. B. Cucumber pickle. Cucumber pickle is a fermentation product of fresh cucumbers. Several lactic acid bacteria are involved in preparation of this pickle. Lactobacillus plantarum is the most important organism required for fermentation of cucumber pickle. 4.6.3 Fermented soybean products. A. Tempeh. Tempeh is a highly popular soybean preparation in Indonesia. The chief organism in this preparation is the mold Rhizopus oligosporus. The boiled soybean seeds are mashed and wrapped in banana leaves or kept in boxes or hollow tubes. It is inoculated with spores of tempeh fungus by addition of a portion of previous batch and allowed to ferment for about 20 hours at a temperature 32 oC until there is a good growing mycelium but little sporulation. It is then sliced and prepared as per the taste, such as roasting or frying. The taste of the tempeh is considered to be bland, but it is highly nutritious. B. Soya sauce. Soya sauce is a very popular preparation of Japan, which has received wide acceptance world over. This is prepared by inoculating Aspergillus oryzae, A. SOA, in a mixture of soaked and steamed soybean with roasted wheat in the ratio of 2:1. The mixture is incubated at 25 to 300 C for a period of 3 to 5 days. Subsequently, it is subjected to various processing steps using bacterium Lactobacillus delbrueckii and the yeast Saccharomyces roxi. After 3 months, the final product is filtered, pasteurized and bottled for use. C. Miso. It is made from fermented soybeans and is a thick paste-like substance. Miso is brownish in color and tastes extremely salty and tangy on its own. Saccharomyces roxi and Torulopsis are the yeasts, and Pediococcus halophilus and Streptococcus faecalis are the bacteria principally involved in miso fermentation. While the most common use of miso is in Japanese-style miso soup recipes, miso also adds a unique burst of flavor to salad dressings, sauces and marinades, baked tofu, or vegetable dishes. 4.6.4 Fermented dairy products. The fermented dairy products assume greater importance in the human diets as, invariably most of the diet consists of milk products, especially the cheese, butter, yogurt, curd, etc. There are a number of fermented dairy products as shown below. Table 4.2 Types of fermented dairy products. Look at the screen. 4.6.5 Economically important fermentation products. The growth of microorganisms or other cells results in a wide range of products. Each culture operation has one or few set objectives. The process has to be monitored carefully and continuously to maintain the precise conditions needed and recover optimum levels of products. Accordingly, fermentation processes aim at one or more of the following: A. Production of cells, biomass, such as yeasts. B. Extraction of metabolic products such amino acids, proteins, including enzymes, vitamins, alcohol, etc., for human and or animal consumption or industrial use such as fertilizer production. C. Modification of compounds through the mediation of elicitors or through biotransformation. And D. Production of recombinant products. A. Microbial biomass. Microbial biomass is produced commercially as single-cell protein, SCP, using such unicellular algae as species of chlorella or spirulina for human or animal consumption, or viable yeast cells needed for the baking industry, which was also used as human feed at one time. Bacterial biomass is used as animal feed. The biomass of Fusarium graminearum is also produced for a similar use. B. Microbial metabolites. I. Primary metabolites. During the log or exponential phase, organisms produce a variety of substances that are essential for their growth, such as nucleotides, nucleic acids, amino acids, proteins, carbohydrates, lipids, etc., or byproducts of energy-yielding metabolism, such as ethanol, acetone, butanol, etc. This phase is described as the trop phase, and the products are usually called primary metabolites. Commercial examples of such products are given in Table 4.3. Table 4.3 Examples of commercially produced primary metabolites. 2. Secondary metabolites. Organisms produce a number of products in addition to the primary metabolites. The microbial growth phase, during which products that have no obvious role in metabolism of the synthesizing culture organisms are produced, is called the idiophase, and these products are called secondary metabolites. In reality, the distinction between the primary and secondary metabolites is not a straight-jacket situation. Many secondary metabolites are produced from intermediates and end products of secondary metabolism. Some, like the Enterobacteriaceae, do not undergo secondary metabolism. Examples of secondary metabolites are given in Table 4.4. Table 4.4 Examples of commercially produced secondary metabolites. Metabolite, species significance, penicillin chrysogenum antibiotic, erythromycin streptomyces erythrus antibiotic, streptomyces griseus antibiotic, cephalosporium acrimonium antibiotic, grisofulvin grisofulvin antifungal antibiotic, cyclosporin A. tolipocladium inflatum immunosuppressant. 3. Production of enzymes. Industrial production of enzymes is needed for the commercial production of food and beverages. Enzymes are also used in clinical or industrial analysis, and now they are even added to washing powders, cellulase, protease, lipase. Enzymes may be produced by microbial, plant or animal cultures. Even plant and animal enzymes can be produced by microbial fermentation through techniques of genetic manipulations. While most enzymes are produced in the trophophase, some like the amylases by Bacillus stearothermophilus are produced in the idiophase and hence are secondary metabolites. Examples of enzymes produced through fermentation processes are given in Table 4.5. Table 4.5 Examples of commercially produced enzymes: organism enzyme, Aspergillus oryzae amylases, Aspergillus niger glucoamylase, Trichoderma ricicellulase, Saccharomyces cerevisiae invertase, Cluisveromyces fragilis lactase, Saccharomycopsis lipolytica lipase, Aspergillus species pectinases and proteases, Bacillus species proteases, Mucor pusillus microbial rennet, Mucor mehe microbial rennet. 4.7 Fermented foods as functional foods. Dr. Elias Mechnikov, a Nobel laureate, around 100 years ago proposed that lactic acid bacteria can render a great service in the fight against intestinal putrefaction and might postpone and ameliorate old age. This concept was developed further through the decades, and today, the emerging probiotics, prebiotics and symbiotics era is a subject of scientific debate and intense research. The use of probiotics, prebiotics, and symbiotics is a promising area for the development of functional foods. Functional foods are generally characterized as foods similar in appearance to conventional foods consumed as part of a usual diet and providing health-related benefits beyond meeting basic nutritional needs. A food can be considered naturally functional if it contains a food component that affects one or more targeted functions in a beneficial way. Dairy foods can be included in the functional food category because of their content of calcium. Specific health-enhancing proteins, conjugated linoleic acid, sphingolipids, butyric acid, and probiotic cultures. Dairy foods appear to be the preferred medium for introducing probiotic bacteria, such as human-derived species of lactic acid bacteria. Example: L. acidophilus, L. casei, L. gusseri, L. rhamnosus, L. reuteri, Bifidobacterium bifidum, B. breve, B. infantis and B. longum. Lactobacillus spp, naturally found in the human small intestine, and various Bifidobacterium spp, a major organism in the human large intestine, are the most commonly used probiotic cultures. 4.7.1 Probiotics. The term probiotic means for life is derived from the Greek language. It is now defined as live microorganisms which, when administered in adequate amounts, confer a health benefit on the host. The most common probiotic organisms clinically useful include members of the bacteria such as Lactobacillus and Bifidobacteria species and some selected strains of Streptococcus, Lactococcus, and members of yeast and molds such as Saccharomyces, Aspergillus, Acanthosis, and Candida species. 4.7.2 Prebiotics. The term prebiotic is defined as short-chained carbohydrates that are indigestible by human enzymes in the gut, gastrointestinal tract, and selectively stimulate the growth and activity of specific species of bacteria in the gut, usually Bifidobacteria and Lactobacilli, with benefits to health. The most commonly used prebiotics in supplements are fructooligosaccharides, FOS. Bifidobacteria, due to the presence of beta-fructofuranosidase enzyme, are liable to break down and utilize FOS. This helps in stimulation of Bifidobacterium growth in the gut. FOS exhibits nutritional properties on colonic pH and stool bulking. It also increases bioavailability of essential minerals and decreases serum triglycerides. The other types of prebiotic substrates include times xylitol, sorbitol, mannitol, times disaccharides lactulose, lactylol, times oligosaccharides raffinose, sorbine, palatinose, isomaltose, lactosucrose, times polysaccharides inulin, resistant starch. 4.7.3 Symbiotics. The term symbiotic is used when a product contains both probiotics and prebiotics. Since the word alludes to synergism, this term should be reserved for products in which the prebiotic compound selectively favors the probiotic compound, example, FOS in combination with strains such as Bifidobacterium B. infantis, B. longum, etc. Combining probiotics with prebiotics could improve the survival of the bacteria crossing the upper part of the gut, thus enhancing their effects in the large bowel. Moreover, the local and the systemic beneficial effects of probiotics and prebiotics might be additive or even synergistic. 4.7.4 Use of probiotics. As per definition of probiotics, the live microorganisms that are administered have to be in adequate amounts to confer a health benefit to the host. Clinical bodies have used probiotic levels of 1 billion to 10 billion or above dosage, according to Earl Mindell, an internationally recognized expert on nutrition. Healthy person should take 2 to 5 billion CFUs, colony-stimulating units, of probiotics a day, and those with GI conditions can take up to 10 billion CFUs per day. In acute, infectious diarrhea, lactobacillus is most effective at a dose of 10 billion CFUs during the first 48 hours, which translates to 5 billion CFUs per day. For prescription, probiotics, the current daily intake recommended is 5 to 10 billion CFUs per day. Capsules and sachets of probiotic plus prebiotic combination, ProWEL, and probiotic alone, Darolac, are commercially available. Benefits offered by probiotic and prebiotic combination formula are times maximum colony forming units, CFUs ensure complete action, times fructooligosaccharide, FOS offers nutrition to the probiotics and normal intestinal flora, times acid-resistant cells, reach intestine in full force, times freeze-dried and nitrogen-flushed cells, offers excellent stability, times vegetable capsules, ensure universal appeal. 4.7.5 Health benefits of probiotics. Few well-designed, well-conducted human clinical trials of probiotics have been conducted over the past 30 years. Only in recent years has the importance of choosing probiotic strains of demonstrated efficacy been recognized. With respect to health benefits of probiotics, research studies indicate the following: 1. Improved intestinal health. 2. Modulation of the immune response and immune function. 3. Reduced risk of cancer. 4. Reduced risk of heart disease. 5. Improved tolerance to milk. 6. Reduce food allergies. 7. Reduce ulcers by decreasing the growth of ulcer-inducing bacteria, i.e., Helicobacter pylori. Fig. 4.3 Beneficial health effects claimed for probiotics. Look at the screen. 4.8 Summary. Fermentation is a relatively efficient, low-energy preservation process which increases the shelf life and decreases the need for refrigeration or other forms of food preservation technologies. It is therefore a highly appropriate technique for use in developing countries and remote areas where access to sophisticated equipment is limited. Fermented foods are popular throughout the world and in some regions make a significant contribution to the diet of millions of individuals. In Asia, the preparation of fermented foods is a widespread tradition. The fermented products supply protein, minerals, and other nutrients that add variety and nutritional fortification to otherwise starchy and bland diets. For instance, soy sauce is consumed throughout the world and is a fundamental ingredient in diets from Indonesia to Japan. Over 1 billion liters are produced each year in Japan alone, although fermentation of foods has been in use for thousands of years. It is likely that the underlying microbial and enzymatic processes responsible for the transformations were largely unknown. It is only recently that there has been a development in the understanding of these processes and their adaptation for commercialization. There is tremendous scope and potential for the use of microorganisms towards meeting the growing world demand for food through efficient utilization of available natural food and feedstocks and the transformation of waste materials. Genetic improvement of the organism is fundamental to the success of fermentation technology. Mutation and recombination are the two ways to meet this end. The alarming increase in inappropriate use of antibiotics and enhanced bacterial resistance, along with renewed interest in ecological methods to prevent infections, makes probiotics, prebiotics and symbiotics a very interesting field for research. Evidence is beginning to accumulate, describing their beneficial effects in a variety of GI, gastrointestinal, and non-GI disorders. They offer dietary means to support the balance of the intestinal flora. They may be used to counteract local immunological dysfunction, to stabilize the intestinal barrier function, to prevent infectious succession of pathogenic microorganisms and to influence intestinal metabolism. Looking ahead, this field holds immense promise for the future in delivering novel therapies in different fields. Thank you. Subscribe to our channel for more updates, and we will see you with the next chapter.